Hi guys, welcome, welcome. I'm so excited to be able to chat with you guys tonight. Looks like we have a bunch of people registered, which is awesome. Although I just read that someone said there's 1,400 people in the AP Gov or something review, AP Human Government or Human Geography, sorry, which is insane. So, but this is our final week, y'all. So I know that a ton of you obviously have other APs that you might be taking this week. So I appreciate that you're carving out some time for bio. Obviously, we're meeting twice during the week, and then we have that big cram session on Sunday, which is free for you guys. I saw some chat about that as well. If you're not a cram member, or if you weren't a former plus member, then you do have to pay $5 for it. But you guys are all crammers, so you get it for free, which is awesome. I have my headphones on tonight, so my sound quality should be better, and we shouldn't have any of those demon noises. Okay, so let's get started. So as you guys know, as I've been prefacing, tonight we're going to talk about FRQs. The two that I picked aren't necessarily directly aligned to units 1 through 3, but I think they're really, really important. Mostly because they talk about standard error, and they're probably pretty similar to what I would expect you seeing on that first big free response. So that's what I'm trying to prepare you for, obviously. We have one last free session at 8 p.m. on this Wednesday, for biology. I believe it's just a question and answer, but I'm going to be streaming at that same time. So just be aware that I'm going to be having that cram. So I would join that just because you guys are all crammers, and I'll be running a session then as well. So Wednesday, 8 p.m., we'll do more FRQ review, specifically tailored to units 4 through 6. And then we have that final cram session from 4 to 9 p.m. I sent out the schedule in the email, but from 4 to 6:30, we have some amazing students who are going to be running, who both received 5s last year, who are going to be running some review guides, some Kahoots, all sorts of great things, going through some FRQs. Then there'll be a 30-minute trivia break, and then I'll be on for the last 2 hours. So I'll be on from 7 to 9 Eastern, obviously. If you guys have any final questions or want to go through a final few things. And hopefully we have less than 1,400 people, just so that people can get their questions answered. But I guess we'll see. All right. Always follow us on social media to get all the updates. And we're just doing FRQs tonight. So pretty straightforward. I see I already have a question. Since College Board says to know evolution, which unit of the units tested on is most likely to have to do with evolution? Just trying to focus on 1 or 2 units especially. When did College Board say? So Lee, I'm not positive. I haven't heard that College Board said you needed to know evolution. Evolution is technically Unit 8, which is not on your exam. There is one small bit of evolution at the end of Unit 5, I believe it is, where they talk a bit about variation. So I would say if you're going to focus on any unit that has to do with evolution, maybe Unit 5. Yeah, as my best answer. But as far as I know, the majority of evolution you do not need to know because it's Unit 8. Unit 7. I apologize. Unit 8 is ecology. So neither of those are going to be on your exam. Okay. Just a reminder, I just wanted to review, I know I did this last week too, but in case you weren't there, just need a refresher, you have 2 FRQ questions, okay? Since we're getting crunch time, let's go through the details really quick. So you have 25 minutes to write for question 1. So that includes reading through the question. You can do scratch notes, which is really great. You can have, like, I would have blank pieces of paper next to me to take some scratch notes, maybe start jotting down some ideas as you're reading through the question. Like, ooh, part A, I want to write about this. Okay, cool. Part B, oh, that reminds me, I want to add this for part A. So I like to have some sort of organizer next to me, so you can certainly have that. And then you'll have a 5-minute window in which you can upload your response, okay? I believe if you start uploading during those 5 minutes and something goes wrong, you'll have extra time to upload, but you should prepare to have those 5 minutes. And you can, I think, type directly into the box, upload a Word document or upload a photo of your writing. So if you would rather handwrite, if that's faster for you, I'm a much faster typer and I like being able to quickly go back and edit things. So that's what I would do, but it's really personal preference. I can't tell you what to do because, you know, I don't know how well you type or if you like to write. Then you have 15 minutes for question 2 with another 5 minutes to upload. And again, as long as you start during that window, you should be good to go. If for some reason something crazy happens with technology, there's a makeup date in June, but we know that's not going to happen to anyone here, and everyone's going to be able to get their scores in or their tests in a week, 1 week from today. So obviously this first question takes a longer time. Whew, sorry guys, it's worth more. It's worth 65%. And these are the questions that we're going to be practicing tonight. These long questions that have to do with interpreting and evaluating experimental results. So I have 2 different experiments that we're going to take a look at tonight that we're going to practice with. As a reminder, you're going to need to be able to look at a scenario accompanied by data in a table and or graph. You're not creating any tables or graphs, okay? So we're going to see that in a second and I'll show you. But you do need to be able to describe the process that is being presented to you, identify experimental design and procedures. So that includes all your variables, which we'll practice with today. Analyze data, which we're going to practice heavily today, and then make and justify predictions based on the information provided to you. Okay, so this question is your big money maker, right? Worth 65%. So that's what we're going to be practicing tonight. And then I'll find some of these shorter ones for us to practice on Wednesday. Okay, so hopefully that works for everyone. It should be pretty beneficial, hopefully. I know usually students really like to do these practice FRQs, so hopefully that is true for you. As a reminder, here are your power words and what they mean. We went over them last week, so I'm not going to go over them again today, but you can read through them if you need to. And we're going to get started with this really lovely table. Oh my God. I'm sorry, guys. We had crazy weather today. It was hailing, all sorts of stuff, and it was like cloudy. So it was just a sleepy Monday for me. And again, not talking to people. So whenever I do these, it makes me yawn. But we're going to take a look at this long FRQ from 2017. Okay, there's a table. Obviously, we need to analyze this. So what that means is we need to become really familiar with what plus or minus 2 standard errors of the mean means, because that's what all these numbers are. So we'll go through that. We're going to read through the question, and then there are some parts that follow. Okay, so I'm going to read through the question with you. As I talked about last week, I know you can all read, trust me. But as I talked about last week, I like reading through these with you guys just because I show you like what words I might skip over or what I would focus on when I'm reading through this the first time, and just kind of mimic how I would attack this problem so that you guys can see and take something from it or not. You have to do what's best for you in the end, okay? But the first thing I would probably look at is the fact that we have a really big old title up here for our table, and it's the effect of caffeine on memory in bees, okay? Notice I skipped over this because the amount doesn't matter, okay? We just have the effect of caffeine on memory in bees, okay? So always look at your table and your graph titles, okay? Because they are a plethora of information because they tell you exactly what you're looking at. So don't look past those. So that's where I would start with this question. Then I would go down to what it says below. So it says, in flowering plants, pollination is a process that leads to the fertilization of an egg and the production of seeds. Some flowers attract pollinators, such as bees, using visual and chemical cues. Dear Lord. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower and use it to make honey for the colony. Nectar contains sugar, but certain plants also produce caffeine in the nectar. Did you guys know that? I didn't know that until I read this question in 2017. Caffeine is a bitter tasting compound that can be toxic to insects at high concentrations. To investigate the role of caffeine in nectar, a group of researchers studied the effect of 0.1 blank, I would just skip over that amount because it doesn't matter, caffeine on bee behavior. The results of an experiment to test the effect of caffeine on bees' memory of a nectar source are shown in Table 1. Okay, so now I'm looking up here. I see we have a control group. We have a caffeine group. We have the probability of revisiting a nectar source plus or minus 2 standard errors, which we're going to talk about in a second. And then we have 2 different time frames. So we have a lot of information in this table that we're going to need to digest before we can answer questions about this. Yeah, so we're going through this and going to figure out what this means as far as if caffeine actually does have an effect on the probability of visiting a nectar source. So some of you may be super familiar with standard error and some of you may not. If I had to guess, I would bet heavily that standard error is going to be in either the table or the graph that you're looking at for question 1. I cannot imagine it would not be there because it makes this a more complicated problem. And because they aren't giving you the responsibility for graphing something, they're going to want to make sure that you understand what you would be graphing or what you're analyzing to the highest level, okay? So we're going to go over that very, very detailed right now because I just have a very strong feeling that that's going to be on your exam. So I'm actually going to switch out of this. So let me just send you guys the link really quick. Sorry. And these slides, the resource should already be attached for you guys. Hopefully it is. If it's not, I'll add it afterwards. But I think I was prepared and put it in advance so that you guys could progress through the slides. And we're going to talk about confidence intervals as well, okay? For sure, because this is some high-level stuff. Okay, so I'm switching over. Okay, cool. So here we go. And I'm just going to show you a basic, let me make this bigger. Okay, so you guys have access. So this is question 1 from 2017. I just sent you guys the PDF. That's probably the easiest way to see this. But I just want to show you guys what a standard error means. So first and foremost, let me just explain what standard error is. So it has to do with confidence intervals, which Maddie says. And basically, some statistician somewhere came up with this calculation in which you can figure out how well your mean actually represents the data that you intend it to represent. Okay, so hear me out. You find a mean, you find an average, okay? And that mean represents a set of data. And you're saying this is the average of all of these data points, right? All of them are unique in their own special way, but we're using one representative. It would be like a class representative. You vote this person in because you think they represent your entire class, okay? That's what a mean does, right? But depending on your data set, some means are better than others at representing that data set. If you have a really widespread data set, it's possible that that mean actually doesn't really represent a lot of the higher or lower end data points, right? Maybe you don't have any in between numbers. You just have really high and really low data. So that mean that's somewhere in the middle isn't super representative of that data set. That's where we have this thing called standard error, okay? It's basically, and again, you don't need to understand the math behind it, but you do need to understand what it means. It is a numeric representation of how accurate our mean is. So if that number is really, really high, it means we have a lot of error and our mean's not doing a very good job at being a mean, okay? If that number is very, very low, it's because that mean is actually very representative of the data set that we're averaging, okay? And so there's very little error. We're pretty sure that the average is what we calculated it to be. And I know that's weird to think about because you just think we calculate an average and it's the average, and that's super fair, okay? But once you get into statistics, we actually realize that that average may or may not be accurate, okay? And so that's what standard error is telling us, and it gives us a range of averages, of means. Sorry, I always say average just because it makes more sense in my brain, but we have a range of means, okay, that we are kind of sure about, and I'm going to explain that more when I draw it out for you. Okay, cool. So if you're taking a look at the PowerPoint slides, I just want to show you, let's just compare our 10 minutes. So 10 minutes for control and caffeine, okay? So what you need to know is that two standard errors, plus or minus, two standard errors, okay? Two standard errors of the mean gives us a 95% confidence interval. Okay, that means if we take our mean and we add 2 of these standard errors to it, and we subtract 2 of these standard errors to it, we are 95% sure that that range that we just created includes our actual mean, the mean that would perfectly represent this data set, okay? This is some weird stuff. So as I keep explaining it, hopefully it makes some more sense. I really want you guys to feel strong about this though. If it's the only thing that you feel really strong about at the end of this session, I'm happy because I'm so sure it's going to be on this exam for you guys, okay? It's sounding like you guys don't have the link to the slides, so I'm just going to send you the actual link. Maybe I didn't publish it. So I'm just sending that out because I know some of you guys are asking about it. Okay, so that's something that you need to know. So in bio, we are always going to do plus or minus 2 standard errors, okay? So in the example that we're looking at right here, they tell you up at the top where it says memory average probability of revisiting a nectar source plus or minus 2 standard errors, they're telling you that the number that they're giving you in that table is 2 standard errors, okay? Sometimes they just give you a single standard error and you need to multiply by 2. So you need to be looking at your source and reading what it's giving you. This one I know is already plus or minus 2. So what I'm seeing at 10 minutes for my control, For my control at 10 minutes, the probability of a bee revisiting a nectar source is 0.72 plus or minus 0.09. Okay, so there's a 72% chance this bee is returning to this single source, plus or minus 9% is what that means, okay? And we'll make a range out of that in a second, okay? For my caffeine group, we have a 0.83 plus or minus 0.07 chance, okay? So this number here, the 72% and 83%, those numbers are our means, okay? So they did this with a bunch of different bees and they found that on average, 72% of the time, bees in the control group came back to their nectar source, and on average, 83% of the time, bees in the caffeine group returned to their nectar source, okay? That's what they found. And up until you took AP Bio, that was enough. And so you probably looked at these numbers and you were like, hey, look at that. The caffeinated bees go back to their nectar source 11% more than bees that don't have caffeine, okay? So you would say caffeine increases the probability of a bee returning to its nectar source, okay? And that would be that, and you would get on with your life. But unfortunately, you decided to take AP Bio, and there's this lovely thing called 2 standard errors of the mean. This will come up again in college as well, and so we need to account for that. And so this mean in and of itself isn't going to be enough. So this is where standard error bars come in. This is where confidence intervals come in. And I'm going to try to explain all of that right now in the context of this question. Okay, so what I'm going to do, and remember that in the question, it already told me that this was 2 standard errors. So I don't need to do anything with this number except for add it and subtract it from 0.72. And that's going to give me my range. Okay, so on the low end of this range, I'm going to have 0.63. Okay, and I'll explain what this means in a second, but that's 0.72 minus 0.09. Okay, so that's how I got that number. 0.72 minus 0.09. Okay, on the top end, I'm going to add 0.09 because it's plus or minus, and I have 0.72 plus 0.09. Okay, and I'll have 0.81. Okay. So what this means is with 95% confidence, because this is a confidence interval, our mean for our control bees is somewhere between 63 and 81. Okay, so somewhere between 63 and 81% of the time would be a representative mean for how frequently these control bees go back to their nectar source. Okay, and this is going to come into play with a graph as well. And with analyzing whether or not data is significant, okay? So let's do caffeine now. So we're going to do 0.83 minus 0.07. So that's going to give us 0.76, okay? And then we're going to do 0.83 plus 0.07, okay? And we're going to get 0.90. So that means we are 95% confident that the average amount of time that caffeinated bees return to their nectar source is somewhere between 76 and 90% of the time. Okay. Our best guess is that it's 83% of the time, but it actually could be anywhere between 76 and 90% of the time with 95% confidence. Okay. All right. I'm seeing some okay, so I'm seeing some questions, so let me just get to those first. So Kendra asks, is there a possibility we will have to calculate standard error or chi-square on the exam? Standard error you will not need to calculate, but you will need to interpret it. Chi-square you very well may need to calculate, and we go over that in last session. So definitely watch that review, Kendra, or take a look at the email that I sent out that recaps, because I have an entire chi-squared screencast from a year or two ago that's posted in there. So take a look at that. Should we have FRQs open today? Yes, Arjun. You should be looking at the 2017 ones. And then can you explain why confidence is important? So the confidence interval just means that if we want to prove this, right? So all of our statistical analysis, like chi-square or like a confidence interval or like standard error, is just we want to prove that our data is significantly different from between our two groups, between our control group and our caffeine group in this example. So if we just look at the mean that we calculated, we would say, yeah, absolutely it is. Caffeinated bees go back to their source 11% more. But now we're going to look at these confidence intervals and we're going to notice that they actually overlap. So that's going to tell us that there actually isn't statistically significant difference between controlled bees and caffeinated bees. And so if we were to publish this in a journal, we would not be able to say that there's a statistically significant difference between these two bees right now, even though to the human eye, it looks like there's a pretty big difference. So confidence intervals just prove to us statistically the difference between our variables. And that's the same thing with chi-square. They're just proving whether or not the differences that we see are small enough or big enough to be considered significant. Besides graphing, will you answer anything with standard errors? Yes, absolutely. And we'll talk about that in a second, Arjun. Okay, good questions. Let's go back to this really quick so I can show you that these two groups are not statistically significantly different. Wow, what a mouthful. It's going to be a long bit of me talking. I apologize. William asked, so if they overlap even a little, there's no significant difference. That's correct, William. If you are looking at this statistically, they're overlapping just a little bit, but it's not a statistically significant difference. So I'm not going to draw out a whole perfectly done graph, but if you were graphing this, which you don't have to, thankfully, you graph your mean. So I would graph like my control group. So this would be 0.72. Okay. And then we would obviously see our caffeine group is a good amount higher at 0.83. Okay. And this is what you're used to doing. And you're used to saying that the caffeinated bees are higher. Okay. When we add these standard error bars, that's where we're going to draw up and down to these two numbers. So I'm going to go draw down to 0.63 and I would draw up to 0.81. And this is the range from this point to this point that my mean could be, but we're predicting it to be right here in the middle, okay? Then on the flip side, we're gonna go down to 0.76 here and up to 0.9. And I know that obviously this is not perfect because I'm just doing it randomly, but this is our range. And that's the mean that we're predicting. That's 0.83. So you'll notice here, there's an overlapping region, okay, where the range of possible means for my control group overlaps with the range of possible means for my caffeine group. And I can see that up here too, right? In between 0.76 and 0.81, the low end of caffeine and the high end of my control, there's actually a segment of numbers that are the same. And so what that means is we cannot say that these two actually have a statistically significantly different mean, right? Because maybe they both have a mean of 0.80, or maybe they both have a mean of 0.77. We're not sure. We think it's about these numbers, which are definitely different. But once we include the error that might be in our data or how widespread our data points are, we actually find that maybe the mean is the same. It's a small chance, but it's a big enough chance that you can't say that they're statistically different numbers here. So Arjun, to go back to your question, we're going to have to answer something that talks about whether or not there's a big enough difference at 10 minutes between our control group and our caffeinated group. And the answer is no, there's not a difference between our control group and our caffeinated group at 10 minutes. Okay. And you would need to write about that. So that's what I'm predicting you guys will have to do on your FRQs, is make a prediction like that. So you would need to have something out next to you where you could do the plus or minus the standard errors, create those ranges for yourself and see if there's any overlap before you say there's differences or there isn't differences. Okay. So I know this is a lot of information to take in, but we're going to keep practicing with this. So hopefully it gets easier. On this graph, how do you represent the overlapping area? By shading. You don't need to represent it at all, and you won't be constructing a graph. I'm only showing you the graph because I want you to see that overlap, but you would not have to one, shade it, or to create a graph at all on your exam because it's all going to be written. Is it saying that the population mean may fall within that 95% confidence range as opposed to the sample mean, which is given? William, yes, that's exactly what it's saying. So we obviously have the sample mean, but we're not 100% sure that that's representative. So we create this range of means that we're 95% confident, which is like kind of a cop-out. We're like, yeah, we think it's 72%, but it actually could, we're 95% sure it's between 63 and 81%. And you're like, well, where is it? And it's like, well, I don't know. We're trying to figure it out. But that's how we prove that difference. Okay. Should we be familiar with other standard errors of the mean other than 2 standard errors? Angela asks. So your other confidence interval would be 1 standard error, which is 68% confidence. I know statisticians, they're terrible. They come up with these numbers. That's something that you could look up on the exam, though. I have never seen them ask about a 68% confidence interval or 1 standard error. It is always 2 with 95%. Because that's what like scientists would use is 2 standard errors. So I would not spend time on that. If for whatever reason it came up, you could just Google it. Okay, so I want to show you guys then we can make no claim then about this 10-minute group, even though your instinct is telling you that caffeinated Bs return more, okay? We can't make that claim. So what we need to do now is I want you guys to look at the 24 hours, okay? And hopefully the first thing that you notice is the difference between control and caffeine at 24 hours is going to be significant, right? And so I'll rewrite the numbers out here. I'm not going to go into as much detail as I just did, but I'll rewrite the numbers out here just so that you can see what it would look like if it actually was a significant difference. So the numbers that they have at 24 hours as opposed to 10 minutes for our control group of Bs, we have 0.41, because remember, it's been 24 hours now, so it's less likely that they're going to return to the same spot. So there's a 41% chance that they're going to return plus or minus 0.07. Okay, so you find that range. I want you guys to find that range just so you can practice. And then I'm going to write the caffeine down here. There's a 78% chance that they're going back to the same nectar source, plus or minus 0.08. And we want to find the range there. Remember that this is already 2 standard errors, so we don't need to do anything to it besides add it and subtract it to find our high and our low for our range. I'll give you a second to prove to me that these 2 numbers are statistically significantly different. Good. So for here, if we subtract, so remember our low end is we're going to always be subtracting first. If we subtract this, we should get 0.36, right? If we subtract 0.41 minus 0.07, okay? And then on the high end, we should have 0.48, okay? And that's from 0.41 plus 0.07, plus or minus means we're adding and subtracting, okay? So there's our range of means for control. And then we should get 0.07 to 0.86 for our caffeinated group. Okay. So you always want to look at the low end of your one group and the high end of your other, okay? And I see the high end here is 48% and the low end here is 70%. So they are vastly, vastly different from one another. These two data sets are very, very different from one another. So we can say that at 24 hours, there is actually a very statistically significant difference in the ability of the caffeinated bees to find that honey source again, or that nectar source again, I should say. Notice that as I was going through this with you guys, I kept making the numbers real, okay? I was like, the caffeinated bees have a 78% chance of returning to their nectar source after 24 hours. That is how you don't forget what you're doing, because it's super easy to just fly through this math, but then it might not make sense to you at the end. You're like, well, what are these numbers and what does this all mean? So constantly, right, as much as I've repeated these numbers and the frequency and the returning to the nectar source, you guys should be practicing that kind of stuff too, right? We're saying like we're 95% certain that the mean for the caffeinated bees at 24 hours is 70% to 86%. Those are the kind of things that you should be saying to yourself as you're taking this exam, because otherwise these numbers lose meaning, and you're going to have a hard time answering the question if these numbers don't make sense to you. Dow asks, how do I know how to compare 0.48 to 0.70? Well, if it makes more sense to you, you can just look at these two number ranges. So a number somewhere in between 0.36 and 0.48 is never going to overlap with a number between 0.70 and 0.86. So I did the high end of my low group and the low end of my high group, but you can also just think about it logically. This set of numbers, like picture them on a number line, is never going to overlap with our higher end. Or if I graphed this out for you guys, like 0.41 going up to and below versus all the way up here, like there's no overlap, right? This is that low end here and this is the high end here. There's no overlap there, right? So there's no chance that these means are going to be the same because their ranges are statistically different from one another. All right, I'm seeing some questions. So two standard errors just means you add and subtract from the mean, Ida. Yeah, so that's what the plus or minus means. So you're adding the two standard errors to the mean and then you're subtracting it from the mean, and that's how you find your range. Maddie asks, how do we know when to multiply by 2? So you have to look at what's given to you. So I'm actually going to close out of this really quick and go back to the question. So hopefully that makes more sense then. Okay, let me make this bigger. It already tells us that we have plus or minus 2 standard errors. Okay, so we don't need to do anything with those numbers because it's already given to us. We're going to look at another example. Let me show you. Actually, this is not the one I thought it was, but I'm going to find that one for us. There's one, so this tells you plus or minus 2 standard errors, and you can see what that looks like on this graph, and you can see that there's some overlap in some of these data points, even though they look very different from one another. But usually they would tell you if it's just one standard error, and I'm going to find the other example of that because I thought that's the one I put in here, but it's not. But that's okay. So, okay, we just analyzed a lot of data. I actually would, though, take the time to do that. You'll notice that the first question here asks you to construct a graph, and obviously you guys aren't going to be doing that, but you should be doing something similar, like constructing those ranges for yourself so that you can actually analyze this data when it's time to do so. Because look at part B. It said, based on the results, describe the effect of caffeine on each of the following: short-term memory and long-term memory. So you need to be able to put into words how caffeine affects short-term memory versus long-term memory. So I want you guys to think about that for a second. Maybe write it out. I think it's really important to write out what you're thinking just to make sure you're including it all. And then we'll review how you can write. This is also your test of, did you understand everything I just blabbed about for the past 30 minutes, right? Because this is putting it all into words. And then we'll review what the answers are. And we'll move on to parts C and D. But obviously we're skipping through part A. Okay, so I'm seeing some people are writing in, and because this is a describe question, you don't need to explain what you're describing. And I know someone brought this up last session, and a super important reminder, if you have time, definitely explain how you know what you know, okay? But all you needed to say here was that caffeine does not affect short-term memory or memory at 10 minutes. Caffeine increases long-term memory or memory at 24 hours. That's all you needed to say. Because remember, describe is actually very short. You're just explaining what you're seeing. Okay. You don't need to say caffeine does not affect short-term memory because the, standard error bars overlap between the control group and the caffeinated group. You don't need to go into that much detail, but if you have time, I would, just because you never know if they might be looking for a little bit more. But you are totally fine to just say caffeine does not affect short-term memory at 10 minutes, improves long-term memory at 24 hours, 2 points. Okay. So it takes a lot of work to get those 2 points because you need to make sense of those confidence intervals. But as long as you start to feel more comfortable with standard error, that will become easier. And I found the other example that I want to show you guys today. So we'll definitely talk about that. Okay, cool. And Ida asks, before we jump into part C, when it says 2 standard errors, the confidence is always 95% yes, right? So all of these numbers are calculated in a very specific way, and it makes it so that you're always 95% certain with that. So yes, Ida. Joan asks, are FRQs graded 1 point per question or 1 point per bolded word? Usually 1 point per bolded word. So this is a 10-point question. So actually A is deceiving. Anytime you have to construct a graph, it's 3 points, but B, you're describing both of those bullet points. So it's 2 points, but usually it's about a point per bolded word. So Kaya, I'm going to show you what it would look like if they just gave you one SC in another question, but it would just say standard error, usually 1 standard error or 1 SEM, as opposed to 2. But it's not always 2. We need to solve for 2. So I know that might sound a little bit confusing, but if you sit tight, I'm going to show you another question where they actually only give you 1, because these are all super valid and we don't know what you guys are going to see next Monday. So it's good to be prepared for any scenario. So you guys are definitely thinking in the right direction. Again, I know this is a lot to process on a Monday night, but you guys are really doing great with it. I think it's really, really going to pay off. So the amount of time that we're investing in it is going to pay off in the long run because you'll be able to make really good predictions about data. Okay. So part C, we're going to design an experiment using artificial flowers to investigate potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits per bee. Okay, so with the designing of an experiment, you have a lot of freedom here, okay? So I just want you to think, what's one way that we could potentially investigate using artificial flowers the negative effects of increasing caffeine on floral visits. So increasing caffeine is our independent variable, and the number of floral visits would be what we're measuring, or our dependent variable. Then you need to identify the null hypothesis, an appropriate control treatment, and the predicted results. So this is going to be 4 points. 1 for designing the experiment, okay? 1 for the null hypothesis, 1 for the control treatment, and 1 for the predicted results. Okay, remember that a null hypothesis, this is the hint that I'm giving you, means that everything you're seeing is due to chance. So caffeine does not affect floral visits by bees, right? Would be your null hypothesis. Null means nothing, so nothing is affecting anything. So I kind of just gave you a point there. But I want you to take a second because this is the best benefit of doing these sessions, to actually think about this. If you're designing an experiment, what would this look like? And what are these different things going to be? And if you're getting stuck, write yourself a note that that's something that you need to review on your own. Okay, so take a minute or two to try to answer all 4 points of part C. Okay. Yeah, so your experiment, Maddie, can be relatively simple, but you basically just need to identify the null hypothesis, control, and predicted results, and that will pretty much design the experiment for you, right, of something that you might be able to do here. And I actually lied to you, not purposefully, but they actually don't give you a point for designing the experiment. They only give you the point for identifying the null, the control treatment, and the predicted results. So that's what I would focus on. Obviously, though, if you were reading this in an exam, you should go ahead and design an experiment as well. Okay, but that's what you should focus on. Good. You guys are writing up some good stuff already. Keep thinking, and then we'll review what the right answer is and move on to part D. Yes, Vanessa. Well, technically, having the null, the control, and the predicted results is designing the experiment. So, yes and no, I guess. Yes, Dow. The control treatment is going to be the same as the control group. Good. I'm seeing some really good null hypotheses here. Start thinking about those predictions then. William has a great setup. Mhm. You don't have to justify your predictions, Maddie. You are correct. Sometimes you do. If you have time, you certainly can, but you do not need to right now. Good. I'm going to give you guys like 30 more seconds. Joan, that's a great question. I'm going to tell you exactly what the rubric says in a second. So hopefully that answers it. Okay, good. I've seen enough, and hopefully you guys are participating as well, because it's really important that you actually write this stuff down or think about it, just to make sure that you know, as opposed to just being like, oh yeah, I knew that. Okay, make sure you actually know it. So I think I saw every single null hypothesis was correct. And all you needed to say for that null was that increasing caffeine has no effect. And you could actually just leave it at that. The increasing caffeine has no effect, period. Or you could say that it has no effect on the number of floral visits by bees. That would be a little bit more thorough. Okay. But your null, pretty much all the time, is just going to be that your independent variable does not affect your dependent variable. So increasing caffeine, which is your independent variable, does not affect the number of floral visits. So maybe write that down as like your null, because remember, you can have notes next to you, like your null hypothesis cheat sheet. Independent variable does not affect dependent variable. Okay. Control, I saw actually only a few truly right answers here. They really want you to say no caffeine, okay? Because that is a true control group. If you say less caffeine, you're still adding caffeine to those flowers. So, because remember, these are artificial flowers. So that's the key word. So artificial flowers do not naturally have any caffeine in them, unlike some of these ones that they're using in the experiment above, okay? So your truest control group is absolutely no caffeine, because that's what we're going to compare other groups to. You can certainly have varying concentrations of caffeine after that, but your control really needs to say no caffeine, okay? So no independent variable for control, okay? Hopefully you're taking good notes here, okay? And then finally, your predicted results. So your predicted results that could be used to reject the null, okay? So in order to reject the null, we need to say that caffeine does have an effect. Okay, caffeine does have an effect on the frequency of flower visits if we're going to reject the null. So they have two different options here. You can say that the number of floral visits by bees is different at increasing caffeine concentrations, or you can say the number of floral visits by bees is increased as increasing caffeine concentrations, but you can actually just say different, because as long as your experimental group is different enough from your control, your null is rejected. So another option that they give is just the number of floral visits by bees is different than the control. So as long as you don't say that it's going to stay the same, you're going to get this prediction point. So this is pretty much a free point. The only reason you wouldn't get it, again, is if you just said that the results stayed the same. That's the only way you wouldn't get that point. You just need to say that it's different at increasing concentrations. Obviously, using the results from above, you would expect that it would increase. Okay. All right, let's see where we're at. You also could say decreased. Actually, yeah, because we're trying to look at negative effects. So you could say that if it gets to a certain level, it's toxic. So you could say, Ida, that the number of visits is decreased as long as you're saying that it's different. That's a great point. I forgot that they mentioned that. Will the test include a design and experiment? I'm not sure, Rhett. It doesn't look like it. It looks like you'll more be interpreting, but you will probably be expected to identify variables like a control group or a control variable, experimental groups, independent variable, dependent variable, all those things you should definitely be familiar with. And yes, Ida, you can absolutely send me FRQs and I can give you feedback. Okay. Same question as Kaya. Is it important to recognize that it's supposed to be negative effects? It's actually not important, Kaya. And that's a really good point. They really just want you to know that it's going to be different than the control, which has no caffeine here. So you don't need to say decreased or increased. You just need to know that it's different. If you said increased or decreased, you would still get the point because you're referring to the fact that it's different. Okay. Cool. All right. Let's see. I think I answered everyone else's questions. Cool. So let's go to part D then. So part D says researchers found that nectar with caffeine tends to have a lower sugar content than nectar without caffeine. Plants use less energy to produce the caffeine in nectar than they do to produce the sugar in nectar. So propose one benefit to the plant that produces nectar with caffeine and a lower sugar content. Propose one cost to bees that visit the flowers of plants that produce nectar with caffeine and a lower sugar content. So even though this is more of an ecology question, I could still totally see you having to answer something like this. But because it's a little bit more content-based, I'm just going to give you a minute or two to think about this. So one benefit to the plant, one cost to the bees. Pretty straightforward here, okay? And then we're going to move on. I'm going to show you that one standard error example. While you guys are working, I'm going to answer Kendra's question. So Kendra asks, for other similar FRQs, will rubrics probably consistently allow you to just say different? Usually with a prediction, you can predict almost anything and get your point as long as it's different from, what the original was. But I would say it's probably easier to just go ahead and say increased or decreased as opposed to just betting that different is going to be good enough. So it's hard for me to answer that for every single question, unfortunately, but hopefully that helps a little bit. And Angela's asking if we can briefly go over some essential experimental design techniques / vocab and how to better understand graphs and tables and controls. Yeah, so we're probably not going to have time to do that tonight, but that's something I can definitely prepare for us for Wednesday. So I'm going to write a note to myself. You guys know I love my notes. And then hopefully you guys are still working on part D and we can review that in a second. Okay, I'm seeing some really good answers, so I'm going to review this with you guys. So the benefit would be to the plant would be that the plant uses less energy, or the plant stores more energy, or the plant has more energy for other uses. Or you could say that more pollen is transferred, or there's more visits by the pollinators, because we know that caffeine is going to increase that memory of those bees. So you could actually go back to that as well. But those would be your benefit points. Mainly, I would assume most people would write about the storing of the energy or the using less energy. Just make sure that you're not simply saying plants use less energy, okay? Because notice here, plants use less energy to produce. You can't just say plants use less energy as the benefit because it's already given to you in the problem. So you got to make it different, okay? So this is actually a really key point, because I have students do this all the time. They just rewrite something that was in the question, not purposefully, but you want to go back and check, because you actually need to say that they're able to store more energy for another activity, or that they're going to have more pollinators. But you can't just say they use less energy because it's already given to you. So hopefully that makes sense. As far as the cost to the bees, you could say, the bees will visit more flowers. The bees will use more energy, right? Because that's both problematic because we're expending energy in those scenarios. The bees may produce less honey because there's less sugar, or the bees may produce lower quality honey, or they may produce honey that provides less energy. But you can't just say, oh, actually you could just say that they're going to be able to produce less energy for that, because that's not given. Okay. But hopefully it makes sense why you can't say plants use less energy as part of your answer to get points, because it's blatantly stated in the part D of the question. Okay. So that's a good tip for later. Can we say that bees lose energy because they get sugar as nutrients from plants? I don't know if I would say that bees lose energy. I would say that they're going to produce less energy, or they're using more energy by traveling to more plants. I had to make that slightly more specific. Die if they consume too much. Let's see. Can be toxic to insect. Yeah, I guess you could say, I wouldn't say die, but I would say it can be toxic maybe if they consume too much, just because toxic, I don't know. We don't know what that level is. And so it might be better to just use that word instead of saying that they would definitely die. But you could still use something like that for sure. So hopefully that helps answer that. All right, we have 8 minutes left. So I really want to show you guys this other problem. So I'm actually going to hop out and not do the second one that I had planned. We're going to jump to 2014. Oops. I'm all over the place. Ooh, no, don't leave. Ugh. Okay. So here's an example. This was the first question on the, oops, this is the answer key. Ugh. Sorry. With you in 1 second. I just want the questions. Okay. Here they are. Okay. We're going to share again. Hopefully you guys have found something useful in today's session about standard error. Should come into play. Okay. So I actually did a whole thing on this, but I didn't really talk about standard error of the mean when I did it because I didn't have enough time. When I did, it was called the 2015 FRQ review, but I did it on this 2014 one because I think it's an important one. And here's actually a really good example because you can see the different means, okay, or the different data points. You can then see the mean, and then you see the calculated standard error. Again, you will not have to calculate standard error. There is an equation for it on your equation sheet, but you will not have to calculate it. Same with standard deviation, which we can talk about. But that's not going to show up on your exam. So just standard error is what you should be worried about. So a lot of people are asking, how will you know if it's just one? So right here, this just says standard error of the mean, SEM, okay? It does not say plus or minus 2 SE, right? But it does say on the axes provided, so this is another graphing question, which you guys aren't going to graph, but you do need to go through basically the same process. Create an appropriately labeled graph to illustrate the sample means of the 3 populations to within a 95% confidence. Okay, so it specifically uses that 95% confidence, but then it tells you that it's the sample mean plus or minus 2 SEM. And they tell you that this is just SEM. So we're going to plus or minus 2, because 2 times 1 is 2, and all of these standard errors are 1 from each of these means, okay? So if I had to go through this quickly, let me see. I think I've tried this before, but I was going to see if I could copy and paste into my whiteboard, because that would be so handy. Of course, I cannot. But I'm going to just show you really briefly on this because now we have 3 different groups. So what I want you guys to be doing on your own right now, and I'm going to be doing it on this whiteboard in a second, is creating those confidence intervals. Okay, so what are going to be the intervals of means for our 3 groups? And then you can start to determine if any of them are statistically different from one another. Okay, so start working on that. You have your means, you have your standard error, 1 standard error. You need to calculate the 2. Okay. And I pasted, yes. So, oh, sorry guys. I didn't realize you had to look at my head that big and not the document. Sorry. You would multiply by 2 because it's telling you that you need to do the 95% confidence interval, which is plus or minus 2. Okay. So I want you guys to find those mean ranges, and then I'm going to show you mine on my separate sheet. Awesome, guys. These are perfect. Yep, absolutely. So you should have 7 to 11 for population 1, 9 to 13 for population 2, and 12 to 14 for population 3. Lee asks, so we didn't multiply by 2 for the last question, and that's because it told us that it was plus or minus 2 SE already. So you just need to read carefully and see if they're giving you standard error or if they're giving you 2 standard error. Here they're just giving us 1, so we have to multiply by 2 because we need to do plus or minus 2 in order to get our sample mean, okay? So a standard error is in of itself a number, but in order for us to be 95% sure, we need plus or minus 2 standard errors. And in my follow-up email, I'm going to send you guys the actual bell curve of standard error that will explain those confidence intervals a little bit so you can see where it's coming from, okay? Okay. So we multiplied our standard error by 2 because it tells us, so it actually gives you guys, if you're paying attention, it gives you the equation right here, your mean plus or minus 2 standard errors. So 9 plus or minus 2 times 1. So 9 plus 2 is 11. So that's our high end. 9 minus 2 is 7. So our range is 7 to 11. Down here, we have 11 plus or minus 2. So we have 9 to 13. And then here we have 14 plus or minus 2 because it's 2 standard errors. So we have 12 to 16. So those are our ranges. And then when you look at them, you can see which ones have overlap. Okay. Hopefully this makes more sense now. I saw someone said that. Please let it be true. Okay, so here we go. I just wanted to show you my work really quick, right? So we're looking at these ranges, and what I can tell is we have overlap between these 2 ranges, right? Because somewhere between 9 and 11, there's overlap, right? For me, I just look at it like numbers on a number line, right? So if I have 7 to 11 and then I have 9 to 13, they overlap in the middle between 9 and 11, okay? You can also graph it out, whatever you need to do for yourself to visualize the fact that these 2 ranges have some of the same numbers in them, specifically 9, 10, and 11, okay? These 2 ranges have overlap in them, 12 and 13, right? 12 and 13 is in this set, 12 and 13 is in that set. So those numbers are duplicated, so those means are not statistically different. The only populations that are statistically different from one another is population 1 and population 3, because 7 to 11 does not overlap with 12 to 16, right? 7 to 11 on the number line, 12 to 16 on the number line. We have a gap in between 11 and 12, no overlap. So there's no chance that the mean for population 1 and the mean for population 3 are the same, okay? So then when it asks you, based on the sample means and standard errors, identify the two populations that are most likely to have statistically significant differences and justify. You would say population 1 and population 3 because their confidence intervals do not overlap, right? Or because their confidence intervals are different from one another, or because, you know, their mean plus or minus 2 standard errors does not overlap or anything like that. But that's the data that you're going to be using in order to answer that question. So whatever way you need to visualize that, if you need to draw out a number line every single time, if you need to graph it, if you can just look at the range and see where there's overlap and where there's not, whatever you need to do to help works. Okay. So Wednesday, we'll go over even more experimental design stuff like the setups, the vocabulary. We'll go over some more variables. It seems like that's where people want the most help because I feel like you guys are solid on content. So hopefully this like kind of just like the fine details is helpful to you guys. Let me answer these 2 questions. Oh my gosh, Ida, the list of all the famous experiments we should be familiar with. I will absolutely include it in the recap. I do have it set up for you. I forgot to share it tonight. So I will have that list of experiments in the recap tomorrow. That should go out tomorrow morning. Okay. I apologize for the delay. And more of a broad question. What do you think is an efficient way to connect ideas from a variety of concepts together, Angela? I really like concept maps. I mean, I rewrite all of my notes and I try to do it in a condensed time frame so that when I'm rewriting one note, I can fresh in my mind remember something that I rewrote from another section. I don't know, everything ties back to basic biology also. So that's a suggestion. But that's something that we can also talk about more on Wednesday or even in our final cram session, just some ways to make those concepts connect. So I'll definitely put that on the docket. And then Grace, last question. How would you do one of these questions if the question asks for an 85% confidence? They would never ask you for an 85% confidence because that technically doesn't exist. There are 3 confidence intervals. It's 68% confidence, 95% confidence, and 99% confidence that are just statistically proven. So it's +1, +2, +3 standard errors. But the bioexam really just cares about +2 standard errors, which is 95% confidence. So they would never ask you for something like an 85% confidence because it doesn't exist. Okay, so yes. So hopefully this was helpful. Sorry, I'm trying to read and talk and do all these things. And, you know, I'm not that talented. Hopefully you feel a little bit more comfortable with this. I really do think, guys, that this is going to show up. I do think it's a good use of your time. If you're still confused, rewatch this video. We can, you know, try to do an example on your own and send it to me and say, hey, did I do this right? I can send you guys, I mean, you could do this whole problem that we just did from 2014 together. Any of those things, but I'm here to help. So shoot me an email if you need more help. Those recap emails will keep coming. If you're not getting them, email me, Caroline at fiveable.me. And I'll make sure that you're added. But most importantly, just thanks for spending your Monday night with me, guys. I'll see you guys on Wednesday. Final stretch last week. All right, guys, have a great night. Stay safe.